So Dr. Liu's a postdoctoral scientist in the Sadler Lab at the Civil and Environmental Engineering Department here at Rice University, and is an environmental engineer by training. Dr. Liu's research mainly focuses on understanding the fate of pathogens during the process of wastewater treatment to achieve safer operations for the public. Multiple advanced tools and technologies are applied, including both short and long read sequencing, single cell. We'll hear more about that later today, and are implemented to really investigate and track pathogens inside of Houston wastewater. And over the course of the pandemic, and wastewater disease monitoring has drawn increasing attentions, and Esther's really been at the forefront of this, and why she's giving a talk here today, so we can learn a little bit more about what went into this when let's say we cannot always assume a perfect sample, and what are the complexities that this metagenome, this mixture of genomes presents to this problem of genome assembly and analysis. And just to conclude here, Dr. Liu's a postdoc currently where she's working on assessing several experimental analysis technologies specific for wastewater samples, primarily to achieve better signal recovery for downstream bioinformatic analyses and for some direct quantitative analysis such as ddPCR. And with that, I would like to hand it over to Dr. Liu for her presentation on leveraging wastewater genomics for public health surveillance. Take it away.
Thank you, Todd, for the great introduction. And thanks everyone for attending this workshop session. So as Todd introduced before, that the majority of my research focuses on wastewater, which is basically public sewage. There are plenty of public health related information that one can decipher from analyzing wastewater samples. For example, on a day like today, we can all agree that detecting viral infections is very important, not only because of the COVID-19 pandemic, but also because of other circulating diseases that can impact our daily lives, such as influenza, rotavirus, stuff like that. So today I'm going to introduce how we leverage wastewater genomics for public health surveillance with some discussions specifically on antibiotic resistance and SARS-CoV-2. So what is wastewater surveillance? It all starts with a positive person using the restroom. So a large portion of infected persons, both asymptomatic and symptomatic, they can shed viruses and bacteria in their stool, which we define as fecal shedding. So the vast majority of human gut bacteria are commensal initially, meaning they're benign to human under normal conditions, but they can develop resistance due to the selection effect of antibiotics. So antibiotics are taken therapeutically to treat infections caused by pathogenic bacteria, but can also result in resistance development in commensal bacteria, a phenomenon what we call standby selection. In addition, horizontal gene transfer, HGT, is able to convert the commensal bacteria into resistant opportunistic pathogens as well. That is the fecal shedding mechanism for antibiotic resistance, and there's also fundamentals of how we can detect those resistance in our wastewater. And similarly for SARS-CoV-2, the fecal shedding studies have also found around 60% of COVID-19 positive cases also have show positive fecal results. And sometimes fecal shedding can even occur 3 to 5 days prior to typical symptoms manifested in clinical settings. And then their stool enters the sewer network, and the wastewater is transported through the network to wastewater treatment plants, where the wastewater get concentrated and treated like in a systematic way. So the wastewater thus represents a pooled community sample from everyone who goes to the bathroom and contributes to the sewage. Untreated wastewater is collected as it flows into a wastewater treatment plant, and the sample is then sent to our lab for further processing and analysis. So we can then analyze the wastewater sample for the genetic information of the resistant bacteria and viruses using different technologies, including next generation sequencing and qPCR or ddPCR. Note that here, for the sake of time, I skipped many critical steps after sample collection till genomic analysis. For example, wastewater concentration and DNA and RNA extraction, both are very important to recover the diluted, degraded, and fragmentized genetic materials of the disease indicators from wastewater samples. So clinical testing, as we all know, is very precise and specific and accurate to one single patient who is likely symptomatic and waiting to go to be tested in the hospital. But as compared to clinical testing, there are multiple reasons why people are super excited about using wastewater as the future of pandemic prevention. First of all, as I mentioned before, both symptomatic and asymptomatic people can be tested in that way because they can all shed virus and bacteria in their fecal matters. And the wastewater is a pooled sample of the community. So instead of, you can only test one person at a time in a clinical setting, if you can imagine that all people contribute to wastewater, then now we can basically improve the throughput of the testing in that sense. And then wastewater testing also does not depend on people taking and reporting the test results. For example, think of now how many people they can basically just test their COVID results at home, which does not have to be reported at all. And wastewater testing does not depend on someone's health-seeking habits. So after the introduction on the power of wastewater monitoring, I'd like to share with you my motivations to apply wastewater monitoring to study antibiotic resistance in the population and what we think we can do using the tool of genomics to mitigate these concerning public health issues. In the US, more than 2.8 million antimicrobial resistant infections occur every year, and more than 35,000 people die as a result. More and more clinical cases reported infections due to multi-drug resistant superbugs. Those bacteria have become resistant to a large number of antibiotics, some of which are last resort antibiotics. So this is why actions are needed to understand the resistant mechanisms and slow down the spread of the resistance. So far, the research community agrees on certain metrics that can be used to evaluate the risks associated with antibiotic resistant exposure to public health. For instance, pathogenic bacteria carrying clinically relevant antibiotic resistant genes, especially those multi-drug resistant pathogens, are more of a direct risk to the public health as compared to certain environmental bacteria that are usually not pathogenic or human accessible. Furthermore, mobile gene elements, MGEs, which can directly and indirectly facilitate horizontal gene transfer out of the resistant genes among microorganisms, are regarded as another significant factor contributing to the risk of ARG dissemination. Therefore, our goal is to use genomic tools to tackle the black box wastewater and tease out those important genetic information of antibiotic resistant genes to study or to evaluate their risks. One of the most widely methods to analyze antibiotic resistance in wastewater is metagenomics. This method is a culture-independent method because it involves direct sequencing of DNA and RNA extracted from environmental samples, such as wastewater, to characterize microbial communities they're in. This method is often referred to as non-targeted or unbiased metagenomics, as no amplification or any other type of enrichment that was involved during the library prep process. This makes this method really a great tool to screen any potential pathogens or resistant genes in a sample. For example, we have high throughput sequencing. It has greatly increased our knowledge of the diversity and abundance of ARGs in the wastewater samples. However, the information about the ARG carrying species and the genetic context remains poorly understood because of the short read length generated by the sequencing platform. Although assembly of short reads might provide such information, the frequent repetitive sequences flanking the resistant genes carried on the mobile gene elements really hamper the effective assembly of the genetic context of the ARGs. So third generation sequencing, the long read sequencing such as PacBio nanopore technologies can really circumvent this issue to some extent. Long read sequencing can generate long reads that are commonly 40 to 200 times longer than the short reads for wastewater metagenome. Therefore, it can hopefully directly reveal the genetic context of ARGs without reassembly. So we performed a head-to-head evaluation on these two sequencing methods to assess their performance in investigating the bacterial host range of ARGs, as well as the association between those resistant genes and other genes of interest. So here I'm showing you the integrated pipeline that we developed for analyzing the long read and short read sequencing data to profile the ARGs and its host. So let's starting with the long reads that are generated off sequencer directly and without assembly. We directly use the BLAST tool to align the reads against the CAR database, which is the base specifically for antibiotic resistant genes. We also, at the same time, used a tool called RGI, Resistant Genes Identifier. We use that for processing short reads. This tool can be used to predict resistance from protein or nucleotide data based on homology and SNP models. And this tool is developed by the same group of folks who developed and curated the CAR database. And they recommend this tool for processing short reads. And then after BLAST, we got the long reads or the short read sample contexts that are carrying those antibiotic resistant genes. And then to annotate the host for these ARGs, we need to distinguish between chromosomal ARGs and non-chromosomal ARGs. This is because from a perspective of microbiology, we can only confidently call a host for ARGs if those ARGs are present on chromosome, but not on plasmid and other mobile gen elements. So that we use the plasful, which is a tool for assigning the location of those genes. And basically we call the host of these ARGs using centrifuge. And then at the same time, we also look at the associations between ARGs and other genes of interest, such as mobile gen elements, using other database, so that we can get like a more comprehensive profile of those ARGs in the wastewater samples. And here I'm showing you some of the results that we generated. So basically, we first compared long read and short read sequencing in capturing the total ARGs present in the samples. So on the y-axis, I'm showing the ARG relative abundance, which by definition is the copy number of the ARGs detected on the long reads data or sample context normalized to the total gigabasis pairs, just the library size of the sample, so that we can compare the abundance of ARGs across different libraries and across different samples and across the methods. So as we can see here, long-read sequencing data shows significantly higher abundance of ARGs, and it's showing more unique numbers of ARGs. So it seems like long-read sequencing detected a significantly more diverse and abundant ARG profile compared to short-read sequencing, which is surprising because considering that long-read sequencing depth was much shallower, only 10% of that of the short-read sequencing. So one explanation for the higher ARG detection sensitivity of long-read sequencing is its better ability to preserve information from raw reads. So to elaborate here, for short-read sequencing data, only 30% of the raw reads map to the assembled context, indicating a significant read loss during de novo assembly step, which is a common issue for environmental metagenome. And after de novo assembly, only about 0.01% of the assembled context pass through our ARG alignment step filter, further reducing the proportion of the raw reads that get involved in the final ARG identification step. But for the long read sequencing, because we don't have to do any assembly, all the raw reads go directly into the ARG alignment step, and we can see that about 15% of reads can align to the database. But there is also one thing that needs to be considered here is for the long read sequencing, especially for the platform that we're using, which is Nanopore. By the time that I was performing my experiment, the sequencing error is still an issue as compared to like other platforms like Illumina. So that's one thing that we should keep in mind, because that could potentially cause some false positive detection in our sample. Next, we profile the abundance of ARGs assigned to each genetic location by each sequencing method. So on the left panel, I'm showing the ARG relative abundance, but across different genetic locations like plasmid and chromosome. And on the left panel, I'm showing you the relative abundance of ARGs detected by each method across different mobile gen elements, including transposes, inter-1, integries, and recombinase, all of which are very important elements for sharing or transferring ARGs across different bacteria. So we can see here that long-read sequencing showed a greater abundance of ARGs that were associated with every single genetic location and every single mobile gene element as compared to short-read sequencing. More specifically, long-read sequencing showed a significant higher abundance of plasmid-associated ARGs, which is 6-fold higher than that of short-read sequencing has shown, and also a strikingly higher abundance of inter-1 associated ARGs, which is 16 times higher than the short-read sequencing. So it seems like long-read sequencing is more sensitive at picking ARGs at any genetic location. This is consistent with what I mentioned previously about the issues with short-read assembler. So now from the application or engineering perspective, we also use these methods to get information of the resistant pathogen profile for wastewater monitoring so that we can, to some extent, understand the potential resistance crisis that is present in the community. So here we focus on specifically on those detected pathogens which are listed on the WHO's priority list for resistant pathogen surveillance. So in this figure, the antibiotic resistant genes are grouped on the y-axis. They're categorized into different categories, like MDR stands for resistant genes that are coding resistance to multiple different types of antibiotics. Also, we have resistant genes confer resistance to quinolone and also to carbapenem or extended spectrum beta-lactamines. All of these are very important as they're commonly used in our hospitals and clinical treatings. And the color blocks are showing detection of a specific host of these ARGs. For example, the yellow color represents detection of the host by long-read sequencing method only, and the blue blocks are showing the ARG host was detected by short-read sequencing, whereas the red shows that they were detected by both methods. So it's not surprisingly that still the vast majority of the linkages were, the ARG and host linkages were recovered and detected by long-read sequencing method, because most of them are yellow blocks. But in a wastewater sample, both long-read and short-read sequencing method altogether recovered 117 ARG pathogenic species linkages, and most of the ARGs are clinically relevant, and also the pathogens are something like E. coli and Pseudomonas. Those are highly infectious disease, causing infectious diseases. So we believe the result is helpful as it provides information of the recirculating resistant pathogens in the community, and we can potentially design further analysis or experiments to specifically target the detected pathogen using either more high-resolution tools to figure out, for example, the mechanisms of the resistance. So for example, currently in our lab, we are routinely culture those resistant bacteria from wastewater samples, and we plan to couple this method with whole genome sequencing on those colonies that we cultured so that we can get more information about the resistant genes and resistance mechanisms, like how do they confer resistance, for example. So other than the host in the study, we found more than 50% of the ARGs detected in wastewater samples were multi-drug resistant genes, which is quite concerning because that means those genes can confer resistance to different types of antibiotics. And ARGs were frequently associated with mobile gen elements. This suggests that they may be involved in the horizontal gene transfer of ARGs among bacteria. And recent studies revealed the striking prevalence of one specific type of mobile gen elements, which is insertion sequence. And they found those insertion sequence are present in resistant pathogens, and they're relatively consistent with certain ARGs across highly diverse bacterial genotypes, indicating the role of insertion sequence in mediating the horizontal gene transfer of these ARGs among bacteria. Similarly, we also found diverse insertion sequence families that were associated with ARGs in our samples. Particularly based on long-read sequencing data, we found the insertion sequence 6, the IS6 family transposase, was frequently associated with two macrolide resistant genes, namely MSRE and MPHE. In addition, by scanning the contextual information of these long reads, we found the structure of IS6 family and the two ARGs is often present on conjugated plasmids. So this finding is supported by the detection of P4 secretion system and MobQ machinery detected on the same long read that's sitting beside these other elements, because these two elements, MobQ and T4SS, are essential and required for plasmid conjugation or transfer among bacteria. So this overall genetic structure highlights the potential for horizontal gene transfer of these ARGs via the interactions of insertion sequence family and a conjugated plasmid. It's worth noticing that this genetic structure was only recovered based on long-read sequencing data, and all these genetic elements were found so that all of the genetic elements were found simultaneously on the couple of long reads as we observed in our data sets. So this finding further highlights the advantage of using long-read sequencing to reveal the contextual information of the ARGs, which is very likely not able to be recovered via short-read sequencing. So as shown by our ARG study, we can conclude that metagenomic sequencing is a non-targeted or unbiased method, demonstrates its usefulness for screening environmental resistant genes and a resistant host. Therefore, as compared to some target methods such as qPCR or ddPCR, sequencing has some clear advantages for discovery and early detection of viruses and other pathogens, because you don't have to know what you're looking for prior to implementing it. Whereas for targeted approaches, you have to know what you're looking for so that you can use the information for primers and probe design. But one challenge with sequencing is that you can often waste a lot of your sequences on things that you don't care about. So reducing those background noise is particularly important for wastewater surveillance because wastewater consists of an unpredictable mixture of diverse genetic materials originating from bacteria, human, protozoa, viruses, and so on. So we're exploring using enrichment methods to improve our signal-to-noise ratio in sequencing results. And one great example is to use tiled amplicon sequencing to enrich the SARS-CoV-2 genomes before sequencing them. However, it is unclear how quantitative the mutation information are that are generated from sequencing wastewater genomes using this type of approach. Therefore, a direct comparison between targeted amplicon sequencing and RT-ddPCR or other quantitative method for mutation detection using wastewater samples is needed. So in this study, in another study that I'm going to introduce right now, we quantified 5 unique mutations of SARS-CoV-2 using RT-ddPCR and perform a targeted amplicon sequencing in parallel on 547 wastewater samples. So those massive number of wastewater samples were collected over the course of COVID-19 pandemic when the Alpha and Delta variants were predominantly circulating in the US. So we compared the consistency in the approaches in terms of, first of all, detections versus not detection, detection versus no detection, and second, the quantitative information generated by each method. For the quantitative analysis, for example, for each target mutation, we assessed the correlations between the mutation concentration as determined by RT-ddPCR in copies per liter of wastewater and the number of reads containing the mutation as determined by sequencing. And there's other quantitative results for each of these two methods, such as the fraction of SARS-CoV-2 containing the target mutation, or like the allele frequency of the mutation as determined by sequencing, for example. So we first to show the results regarding detections by these two methods for each of the 5 mutations. So here, the 5 mutations are listed right here. And the number on top of each quadrant represents the number of detection events that fall into each detection scenario, denoted on Y and X-axis. So Y-axis is regarding the detections by the tiled amplicon sequencing, and the X-axis is denoted is denoting the detections by RT-ddPCR. D stands for detected, and D stands for not detected. So for example, here we have 164 detection events that were both detected by these two methods, whereas only 66 of detection events are only detected by RT-ddPCR, but not detected by sequencing. So if we highlight all consistent detections, so referring to those that are double detected or double not detected, we can see that they account for a vast majority of all detection events. However, if we highlight all inconsistent mutation detections, referring to those that are only detected by one of the methods, we can see that still it is more likely for an event to be detected by RT-ddPCR as compared to detected by targeted amplicon sequencing. So in fact, when we observed positive mutation detections by RT-ddPCR, about 43% of those detection events were missed by sequencing, meaning sequencing was not able to detect them at all. But when sequencing reported not detected mutation detections, about 30% of those events were instead positive detections by RT-ddPCR. It's all highlighting the relatively poor sensitivity of detection of sequencing as compared to the sensitive RT-ddPCR method. Furthermore, detection via targeted amplicon sequencing could also be significantly affected by the inconsistent or uniform single base coverage across the SARS-CoV-2 genome. Single base coverage is defined as the number of total reads aligned to all the specific genome position. So here I'm showing you the average single base coverage across all 547 samples with respect to the locations on the SARS-CoV-2 genome as shown on the x-axis. So apparently the read coverage along the genome was definitely not uniform, leaving substantial regions with few informative reads. This issue is inevitable for this type of multiplexing PCR-based sequencing because the primers can have different efficiencies of binding. So to elaborate on the impact of dropping single base coverage on mutation detection, here I'm showing the two, for example, the two regions that are amplified by the RTIC V3 primers. So the top panels are showing the regions amplified by the primer 72. The bottom are showing the regions amplified by the primer pair 76. And as you can see here, that the average coverage across the region amplified by 76 is significantly lower as the region of the coverage of the region amplified by the primer 72. So, and because these regions respectively are covering different mutations, one is for deletion 69/70, one is for N501Y. So this amplification bias resulted in less coverage at this specific mutation site as compared to the other one.
Esther?
So this probably leads to, yes.
So I think we're at time now, so if you could just wrap it up, that'd be great. I think we're
Yes.
okay, thanks.
Okay, sounds good. So I'll quickly go over.
Yeah, maybe one or two more slides. Thanks, Esther.
Okay, I will do that. So then we look briefly into the other quantitative matrix generated by these two methods, and we found only slight significant correlations between these two metrics. So then we have our results, which is basically that people should caution the use of quantitative measurements of SARS-CoV-2 mutations in wastewater samples if the results are solely based on targeted amplicon sequencing. But despite its lower sensitivity nature, sequencing still has a clear advantage of being more comprehensive, not limited by a prior knowledge of the target mutations, and enables the discovery of cryptic lineages and emerging lineages of concern discussion. So that we believe in the future that we should really combine these two methods to achieve, first of all, the quantitative confirmation of the targets and also the comprehensive screening of all the mutations present in wastewater samples. So here, I really want to say thank you to all the collaborators involved in all of these projects. We're closely collaborating with Dr. Todd Tringen at Rice University, and kudos to those amazing collaborators without whom none of the work would be possible. And thank you. And with that, I'll take any questions if we still have time.
Great talk, Esther. I think we have time for maybe one question here, and does anyone have a question for speaker? There we go, sorry.
So for the antimicrobial resistance, how do you know you're reaching saturation of detection between the two different platforms? And just that, like, if you keep throwing reads at it, you're going to keep finding more antimicrobial resistance genes, or do you actually reach a plateau in kind of what you're detecting? Like, how much sequencing data do you need to collect to be able to be confident that you've accurately profiled the sample?
That's a great question. I think that normally in our environmental surveillance field, we normally have a convention that, for example, for both of the method, we collected 5 gigabytes of raw data, or I would say 5 to 8 gigabytes of data. That would be relatively enough. Unfortunately, in our study, we didn't do that type of test because it's really how could you determine the threshold for the unknown? Because we don't really know how much resistant genes are present in there. I think if you want to do that type of analysis, maybe we can couple some of the metagenomic sequencing results with some, say, qPCR data to see if that is relatively adequate coverage of the sequencing data that we obtained. But in our study, we didn't do any of these type of testing. That's a good question. We should consider that in our future studies.
All right, Esther, let's thank our speaker again. Thanks, Esther.
Thank you.